Well, I don't know how many of you grow cucurbits. I assume some of you, since you're here. We've been working on trap cropping as a potential method for controlling striped cucumber beetle and cucurbits, squash, etc. So I'll fire up for that. It's going to work. There we go. Now, I'm sure you're familiar with what happens on early season when they do show up mid-June or so. They can obliterate plants. What we see is a lot of them will grow out of it. But if you get hammered early on, you're going to have problems establishing stands and whatnot. So I think it's early season protection that's most critical. Probably literature would suggest the same thing. Everything I've read says later season probably not going to sustain much yield loss if the damage comes late in the season. That's true not only for direct foliar feeding, but also from bacterial wilt that they vector. Don't know much about what goes on with the soil blowing phases and what effect they have on cucurbit. Okay, we had several hypotheses that we wanted to test. And it was based on flowering, essentially, because a lot of research has been done with using, say, blue hover squash to protect something like a butternut squash or a cucumber. And it always seems to work, but we don't really know whether it's going to require some kind of specific pairings. I mean, you got one crop you want to protect, what's going to protect it? And that's kind of mind-boggling when you think about all the permutation. So flowering may be important, and genotype may be important. That is, the genus and the species, not just whether it's flowering or not flowering. And different varieties are going to have a different time of flowering. So if we can determine that flowering really is important, then we can start looking for varieties that flower early and use them as something to aggregate the beetles away from your main crop. So, several trials. We wanted to know whether a certain variety could be used to protect that very same variety based on if we could get it to flower earlier by setting it out as a transplant that's going to flower a couple weeks earlier than the main crop. We wanted to know whether genotype is more important than the flowering, or flowering more important than genotype. We also wanted to know whether there was some varieties that could be used as a generic trap crop based on their flowering. And lastly, we had some add-on work that wasn't funded by the particular foundation that funded this work. And if I have time, I'll speak a little bit about that at the end. Oops. So a basic trap crop, sort of generic design, where you've got traps on the outside, border rows, and your main crop in green on the inside. And on our particular trials, they were about 30-foot plots, give or take. And we didn't plant them in exactly commercial spacing because we wanted to take some data. I think I did that. Okay, about every 3 or 4 days we go out, we count beetles, we count number of flowers, male flowers, female flowers, number of beetles on flowers, number of beetles on foliage. Just a basic monitoring of what beetles are where and where on the plant, whether in the trap crop, main crop, whether on flowers and foliage. Right. So, a few graphs in here, bear with me. What we did find is that it's pretty easy to get your plant flowering. You got a pointer? I got a pointer. If you take a look at the upper figure there, the upper line. Ooh! I'll have to remember that one, though. Maybe you just go back and go forward, reset. Well, I'll tell you about a portion of the graphs at this point. I guess I'll use my little finger on that one from now on. Okay. Looking up here, this red line That's male flowers on the trap crop. And if you look at female flowers down here, way fewer female flowers than there are male flowers. That may be common knowledge, I don't know. It wasn't apparent to me until I started. Now that is Black Beauty zucchini. And over here, this is also Black Beauty zucchini that's been put in later. And if you look at the foliage down there, number of female flowers, they're very few again. So basically it's male dominated in terms of flowering. And those are the ones that really aggregate the beetles just because there are more of them. So we take and kind of smooth those out a bit. You can see the patterns that the early planted Black Beauty had earlier flowers and the later planted Black Beauty. Now if we take a look at the beetles per plant, you can also see that they pretty much were on the flowers. Now this is male and female flowers together, but the beetles were on the flowers in both the trap crop and the main crop. And on the foliage, pretty much nothing. So the beetles are obviously attracted to the flowers, and that may give us a means of, let's see. So that's everything combined at this point, flowers and foliage, male and female flowers and foliage. And the number of beetles is obviously greater in the trap crop that was planted a little bit earlier than the other Black Beauties. Now if we superimpose these, you got to remember how the scales are different here. Flowers is a different scale than beetles. But if you superimpose the flowering pattern with the beetle pattern, there's pretty good agreement, just visually, that the beetles are following the flowers in both cases. So what we conclude from that is that the beetles definitely were on the flowers, mostly on the male flowers, mostly we think because the male flowers are just more prolific than the females. You can actually use the same plant to protect them. The very same plant, as you just separate them in terms of what their ages are when you go in the field. And it again says that maybe we could use just some sort of generic early flowering cucurbit to protect all different kinds of cucurbits. And that would simplify trap cropping quite a bit. We ran another trial also, and that was where we were looking at Blue Hubbard to protect Black Beauty zucchini. Now in this case, the Blue Hubbard also went out earlier than the Black Beauty. It was planted, I guess we got about a 2-week differential on that, something like that. Again, male flowers and female flowers. Obviously, male flowers predominate. When we take a look at the patterns, though, it was a little bit different than Black Beauty versus Black Beauty. Yeah, that's the Black Beauty is the main crop, and the Blue Hubbard is the trap crop in this case. The Blue Hubbard just it just kept flowering. Whereas the Black Beauty, it flowered, and then as the plants got bigger, it just sort of tapered off. That may be in part because we didn't harvest the fruit, and they just quit flowering. But in this case, Black Beauty had more flowers than the Blue Hubbard, but it was a little bit later in the season. And when we look at beetles, again, the same pattern that we saw before is that they're on the flowers, and they're not down here on the foliage very much, except in the early season. Before too much flowering occurred, there was an aggregation of beetles on the Blue Hubbard, preferentially over on Black Beauty. So if we take and just generalize here, foliar, male flower and female flower beetles put them together, we were pretty successful in aggregating them in Blue Hubbard. Now Blue Hubbard has been used against waltham, and it's been used against to protect waltham butternut. It's been used to protect cucumber. And this seems pretty straightforward. It works. But, is a Blue Hubbard a generic crop? And do you want a Blue Hubbard? Because can you get rid of it after you grow it? So anyway, if you superimpose the patterns, it became a little bit interesting because we had, again, Black Beauty in black here was flowering more than the Blue Hubbard was, but we had more beetles early on in the season, especially when we really want to in this region when your plants are quite small. We were able to successfully aggregate them early in the season. I think that's a critical factor. If we have time, we can talk about that some. Again, same conclusions pretty much. Beetles moved to the flowers. It was interesting that the Black Beauty had more flowers and they were still aggregating on the Blue Hubbard. And it does suggest that there might be some genotype involvement, which would suggest that maybe we would not be looking at, say, Cucurbit maximum genotypes as a generic early flowering trap crop, if we can find an early flowering Cucurbit maxima. And there's a guy in Geneva that has 600 or 700 lines of these things that we can take a look at. Okay, a separate trial that we did with Mike Mazurek, who's the cucurbit breeder at Cornell. We took just to see if we had quite a bit of variation in when the different cultivars or experimental lines would flower. It was a simple scientific design. Took a look at flowers weekly, and just took a look at 10 different lines that he had sort of a gut feeling about when they would be flowering. Those are the ones that we chose. Some of them are organic approved, some are not, but you can use them because there's nothing organic in that line, like Golden Arrow, pretty early flowering, it's not organic, but you can use it, apparently. You know, don't pay attention to all lines. The bottom line is that, yeah, there are flowering differentials, where we had Golden Arrow in the blue was pretty early flowering, and one of Michael's cross was Godiva like Costata. That was pretty early flowering. And Success PM, I just thought it was interesting because it flowered for so long and it had so many flowers. So if we could get a plant that has a lot of male flowers and flowered early and was genotypically attractive like a Blue Hubbard, well, and then we have a genetic trap crop that maybe anybody could use to protect any cucurbit. Let's see. Yeah, we weren't able to fulfill some of our funding obligations, so we threw in a little extra work to help use up the money. We took a look at what happened in the soil on the trap crop versus the main crop, and it was not really that informative, but we did see that basically that's the number of soil dwelling phases we saw, we didn't count eggs, they're a little difficult, but we counted larvae, pupae, and adults in the soil, the adults before they emerge. And what we did find was that really the number that we found was not comparable. I mean, it was pretty comparable. There's no difference there statistically, although I don't think I even tested that. What we do see is a shift in life stages where we're finding a few more adults on the trap crop would probably suggest that their beetles are moving in there and ovipositing, laying their eggs earlier, and thus they're developing a little bit faster than in the main crop. And we saw that even more in the phenology trial, which was a Black Beauty with Black Beauty down here. And there was quite a few, there were quite a few more adults than there were in the main crop. The numbers were pretty low, 64 that we found versus 20. It was a different soil, and we just didn't find as many soil dwelling phases at that point. How are we doing on time here? So there were, that was kind of some add-in work we did. And then lastly, this, I don't know if you're going to be totally interested in not in that, but the squash beetle wilt system is pretty interesting ecologically, because the beetles, they aggregate. They aggregate in response to a male-produced aggregation pheromone. But when we tried using this pheromone by itself, you got zilch. I mean, it just didn't attract anything. So previous research has also suggested that it may have to work with a plant volatile. So we're working with a chemical ecologist who looks at plant volatiles and their influence on predators and on herbivores as well. And so when we took Blue Hubbard squash without any beetle damage, we see some interesting peaks there. This particular noatriene has been shown to be induced in cucurbits by the feeding of plant-feeding mites, two-spotted mite, and that particular chemical will attract predaceous mites that eat the two-spotted mite. Now, if you add beetles to the system with the Blue Hubbard, you see a ramp up of this hexanol and a suppression of these sesquiterpenes. And this says basically the beetles are doing something to the plant, and that guild of volatiles that they're sending out changes when there's beetle damage going on. Now, Black Beauty, that's about flatline. You know, that sucker's dead, you know, it doesn't have anything going on. But when you add beetles to the system, you see this triene, and you also see the ramp up of the hexanol and a few other compounds as well. The beetles are really doing something on the Black Beauty. So it's pretty suggestive that the beetles feeding on the plant. So this is the way it works. At least the woman that worked on it has this really simple sort of description for what's going on, is that a male beetle lights on a plant somewhere and says A or nay to the plant. You know, it's good or it's not good. If it likes it, it sends out the pheromone. And that draws in both male and female beetles. And when the males start to aggregate, they send out more pheromone, and more females come in. Now, you know, you look on your field, you see one plant that's just covered with beetles, and the one next to it doesn't have much. So it may be something that we can work on. If we can start breeding for either suppression of some of these volatiles or amplification of them, if we want to use them in the trap crop. So we can do something like a push-pull system where we're breeding to suppress and breeding to amplify, so we push the beetles away from the crop and into another crop where they can be potentially controlled by some approved chemical, which Abby's going to figure out pretty soon which ones work on beetles, right?
No way.
Well, but I hear is nothing works on
That's a brand new one.
There we go.
And I know about's going to work out.
So anyway, that's, I think, getting close to it. Conclusions in general, flowering is real important. The beetles are going to be on the flowers. I almost guarantee that. Early flowering lines, if you can get them out and get them flowering early enough, you know, that may be just a cultural scheduling. Can you put your transplants in early enough, and can you put them out early enough to have some kind of flowering going on? In the absence of that, maybe you can find a genotype that's attractive, like blue Hubbard, that sells better than blue Hubbard does. There's some question about whether the egg laying and development on the soil has an effect on later season damage on the fruit and this and that. That's a bit of a gray area, and I don't think anybody really knows what's going on with that. But you may be able to trap beetles into the trap ground, have them develop there, and then terminate them with some kind of soil tillage, probably mowing and tillage, because mowing alone does not do it. And then if we can figure out some volatile differences between cultivars, we can breed for them, or, I don't know, maybe you can use them like pheromones or something. Maybe they be approved that way, I'm not sure. So this is what we want to do in the future, to working away at this whole trap cropping system. Look at the potential for volatiles and how they relate to aggregation in beetles with the pheromone. We want to look at specifically Cucurbit maxima, which is blue Hubbard types. Look at them as a genotypically attractive line, plus look for early flowering. And then what I'm going to do in the near future is to tease apart this genotype flowering effect, because we're going to go in and we're going to cut flowers off of various things and see where the beetles move with and without flowers. And then if we can determine that trap cropping is simple enough to be implemented, and if we can then attract the beetles to the trap crop, then with some future work, perhaps we can find some chemical control, approved chemical control. Tank mixes, something new, I don't know what's going to come up with that. But that would be the directions that we want to pursue. So Abby helped with this, Abby Seaman. Michael Mazurek was the plant breeder, making suggestions as to what plants to use in flowering differential trials, and we'll be submitting some proposals to do more of this work, also related to downy mildew in cucurbit. And Shaw and Lucy over at Stick and Stone Farm in Ithaca helped us provide a land and whatnot. I think that's all I have. That'd be it.
Any questions for Jeff?
So when you did your trials, did everything get put out as transplants?
We did.
Okay.
Yeah.
Yeah, because the one thing that people seem to like better than flowers is
Very young seeds. Yep. Yeah, I think you have to remember that beetles will kind of, if they have a chance, aggregate. If you can spread them out over the seedlings, they're not going to kill them all. I think one of the appropriate approaches with seedlings is you put in more plants than you need. I think everybody probably knows that. Just some sacrificial losses there. But generally, when plants get hit, if they don't get killed, typically some varieties, much more than others, will grow out of it and produce pretty darn well. And all Michael's work has shown that. Certainly our stuff has shown it with, I think it was Seneca, was the variety we were using on that one, way back when.
Other questions?
I understand that the one lays the egg
question is where do they lay their eggs? They lay their eggs at the base of the plants in the soil. This is in the springtime, June to mid-July. And that's mid-June to mid-July, I should say, at least in our area. Those will develop and emerge as adults, which then feed on cucurbits and acquire cucurbitaceans that they then give as a nuptial gift when they mate the next year. So when you hear about cucurbitaceans being the thing that's attracting beetle, that's kind of questionable. It's probably something besides cucurbitaceans, which are certainly not volatile to begin with. But they can taste them. You can taste them. They're bitter. They don't overwinter in the soil. The adults may.
Okay, the adults.
They leaf litter in the soil, things like that. Yeah. And we don't know whether they leave the field or whether we don't know where they come from at this point.
Okay.
Some of them do stay in the field.
Is there anything that you can do, or anybody is doing research on what you can use in your soil to keep that from overwintering?
I don't
The
know.
question, Jeff?
Hmm?
Repeat the question.
The question is anybody working on ways to prevent them from being in the soil and overwintering there, coming out next spring and initiating the recolonization? I don't think so. We're proposing that in some stuff that's coming up, where we'll take a look at what's coming out in the spring from the field that they started in. And other people have done that, and they do come out from the field that they've started in, but they also come from elsewhere.
Nobody knows really where they're coming from. What you're doing is trying to see what about the flower is attracting them from wherever they're coming from.
Question is, are we trying to figure out what about the flower is attracting them?
Yeah.
No, I think they know that already. It's probably indole. Yeah. And actually there are baits out there made with TIC, which is trimethoxybenzene, indole, and cinnamaldehyde, but they're not organically approved. But other than that, those three compounds, some are more attractive to pollinators, some more attractive to the herbivores like the beetles, and some attract both each equally, sort of. So if you select for flowers that have, say, a higher percentage of indole, you may be able to attract beetles without interfering with pollinators or some permutation there. Yeah.
Questions?
Well, just wondering if there's been any work done on some of the nematode predators working at the base of the plants with the larvae.
I think they're having.
So that might be a way that you could try to reduce the next generation. I don't know if it works.
Right, well, if Jeff can attract them all to one place, maybe we can try the beetles on those.
Well, you know, they did studies in Minnesota, and granted, Minnesota's different here, but the overwintering mortality of beetles in Minnesota is huge. Meaning that probably in New York it's pretty high too, and yet we see bajillions of them every spring. So I don't know where they overwinter, I don't know where they're coming from. But they do feed on all different things. They just have to have a cucurbit to lay their eggs and to mate. But they'll eat pollen and this, and you know, they eat hibiscus or what is it? Okra. I've seen them on okra. You know. So they eat a lot of things, but they obligate to a cucurbit when they're ready to mate and reproduce.